Brownridge, I have a present for you.
That's precious.
Hey, what? I found it on my bedroom floor the other day. It's not mine.
It's a tooth.
Yep. I found it on my bedroom floor and it's not mine. And I decided that you were the best person to give it to.
So if I put it under my pillow, I'm supposed to get money?
Yeah.
Thank you.
You're welcome, Dr. Brandon.
Hemagglutinin.
Because I got it in the gluten and then the minidase when I was with pressure, but that's not involved now. It's just the hemagglutinin.
Both of them are involved, actually.
But the HA, where it says HA, I mean, I was trying. Okay, fine. Fine.
So not going to join via Zoom?
I mean, I would assume that would.
Well, we are starting. Well, if she comes in, then she can. Okay, gang, let's go ahead and get started. On the subject of Jessica, some of you may have heard she had a death in the family. It was a grandparent. I assume she was rather close to that grandparent, and so it's hit her pretty hard emotionally, so she's a bit on the rocks. Shut up! There. Okay, so we're going to go ahead and get started. Keep Jess in mind as we proceed today. And she didn't get a Zoom link.
Yeah. That's why no one's joining me on Zoom.
Okay, there it is. Let's believe they'll join. All right. Okay. All right, here where we left off last time. Okay, here they come. All right. Okay, so last time we took a look at the fusion of the flu virus with the host cell. This organism, well, calling a virus an organism is a stretch. It's not cellular. This entity, shall we say, and they turn the heat on, and my gosh, it is hot in here. So the trimeric receptor is a combination of the hemagglutinin and the neuraminidase. These vary from strain to strain. That's why you have different strains of flu, H1N1, H5N2, H10 to the 6th, and 10 to the 3rd, you know, so on and so forth. And these vary from season to season, and that's why you need to get a flu shot every year in order to make sure you're protected against the particular strains that are coming up. And then H1N1 itself has many, many different substrains. So there's Bangkok, H1N1 Bangkok, and H1N1 London, and H5N2 Beijing, and so on and so forth. So the epidemiologists try to guess which will be ascendant. Sometimes they get it right, sometimes they don't. Even if they're close, getting a flu shot will still cause you to have a less severe flu than even if you get sick. Amanda, go ahead and ask your question.
You mentioned
Snare? Yeah.
what exactly are those?
I'll get to that. Clem? Based on the reservoirs. These are reservoired in animals. They're reservoired in waterfowl because the virus is stable in bodies of water, temperate temperature, and they're also reservoired in swine. So you can sample the animals. It's also very clear that recombination occurs in these reservoir animals. And that recombination can provide new strains because this is a segmented virus. The genome comes in segments of -strand RNA. Andrew? That's a different virus that started in a different critter. You have new strains, of course, that arise. That one, SARS, comes from bats. There's thought that SARS-CoV-2 was then resident in pangolins. That's not been nailed down. That is a good guess based on viruses isolated from pangolins, but that one is still debated. It's possible. Definitely, though, had it in bats. Definitely. So did SARS-MERS, reservoired in camels. So the Middle Eastern Respiratory Syndrome, that one was in camels, disgusting creatures that they are. So those are different groups of viruses in this. This is an orthomyxo virus, negative strand. SARS is a plus strand virus. Different birds, similar feathers, but different birds. Now, this has provided a model system for membrane fusion because for years people thought, well, take one membrane to another, yeah, they're going to fuse. What else are they going to do? Well, in fact, if you experimentally test this, they bounce off each other. So membranes aren't just going to fuse. The cell, in fact, has a machinery that allows membrane to fuse, and the cell also has a machinery for deforming membranes and changing the topology of the membrane. So if you look at the various things inside cells and the structure of these things, you see things like flat discs, and then you see the deformation of the membrane into a vesicle. To be able to deform the membrane, you have to be able to pull it in, pinch it off. These two bits then have to fuse. That's a bit of a trick. Yep. Viral infections, of course, you have fusion of the viral membrane, or if the viral is not enveloped, it's taken up in a vesicle, and then it's uncoated, usually in a lysosome. You have fertilization, fusion of egg and sperm, and then the pronucleus is brought into the cell, and then that genetic material combines with the nuclear material. You then have fusion of various vesicles. It's their plants. That's the case for vacuoles, with vesicle fusion and budding. It's very common in animal cells too. How on earth do you do stuff like this? You have to sort the proteins to an area, and you have to pinch off that area. Turns out there are proteins in cells for deforming membranes and for doing this sort of thing. And you can have proteins, first of all, highly charged, highly positively charged. They can associate with the negative charges of the phospholipids. They can also have little amphipathic helices. They can insert in the membrane, and then when they do that, they can actually exert shear forces that will deform the membrane. And there are proteins of so-called BAR domains, bar domains. Bar domain proteins are known to assemble on membranes and deform the membrane. They do this very effectively. They maintain the curvature of membrane. They put new curves into membranes. All kinds of very cool sorts of things. And proteins like this, they have positive charges here, amphipathic helices that can insert in, and then they can deform the membrane. So this intrinsic curvature of the protein plus its association with the membrane can actually force the membrane into the same topology as we have with that other side of the membrane. Also, if we stick these amphipathic helices into the membrane, because you're going to change one leaflet of the membrane, the other side of the membrane has to change too. So that's why these proteins only need to insert one leaflet thick. You just change this curvature, this will actually just follow suit. And then your VAR domain proteins can essentially favor and maintain curvature in particular membranes. Many of these proteins are now being isolated and characterized at very high resolution. Journal of Cell Biology monthly seems to have a new article on some protein in the Golgi, in the ER that helps maintain flattened discs and curvatures and things like that. Virtually all of these proteins have a marked ability to interact with certain lipids found in those membranes. And what about membrane fusion between vesicles? You have these two things that are charged. They're going to just bounce off each other. Bonk, bonk, bonk. So what gives essentially with these? Well, it turns out that membranes that are going to fuse, and one model system where this has been very, very highly studied is neurons, because neurons have vesicles filled with neurotransmitters. Getting those vesicles to fuse with the membrane of the synapse, in fact, a very good model system. And you can grow neurons in culture. You can get them to synapse with other cells. If you do that, you can then study at some high resolution this model system. And so turns out that the vesicle has a protein that is relatively unstructured. It's called a vesicle snare. A vesicle snare. We're going to call it snare because snare stands for N-ethylmaleimide sensitive factor attachment protein receptor. If you don't mind, I'm just going to call it snare. Is that okay? I thought so. So there's snare, a vesicle N-ethylmaleimide sensitive factor attachment receptor protein receptor. Then the target, the plasma membrane, the whatever, has another structured protein. It is a target snare. And then there's an assistant protein, what they called a SNAP. A SNAP. V is for vesicle, T is for target. Target. SNAP stands for snare cysteine protein. And because biochemists isolated these, they have names that will just absolutely put you to sleep. SNAP1, SNAP2, SNAP3, SNAP4, SNAP5, 6, 7, 8, 9, 10, blah, 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 blah. Oh, did you say something? The snares have much more creative names. Syntactin, Vesculin, all kinds of glorious names. The SNAPs, sorry, don't look for interesting names with the SNAPs, but look for them with the snares. Now, how do you get this sort of thing to work? Because it takes energy. To get this to work, turns out there's a small GTP binding protein that will hydrolyze GTP and is also associated with the membrane. And these particular proteins, they are called RABs. RABs. These are small GTP binding proteins. And when they bind to GTP, their hydrolysis of GTP is going to essentially help with the formation of the complexes that are going to form. So these proteins, as we said, are rather unstructured. However, when a T-snare, a V-snare, and a SNAP get together, they discover that they have hydrophobic sequences that are short and don't like the water. So what do? They wrap around each other to get away from the water. And they form a coil structure between 4 different alpha helices. And this is called a snare complex. A snare complex. And it forms between the SNAP, this is SNAP 25, again, beautifully boring name, and then a V snare and a T snare. And the place again where this has been well studied is between a V snare called VAMP, which stands for vesicle associated membrane protein, VAMP. VAMP is one of the big V snares that you find in neurons. And then its target, which is syntactin. And that's a T snare. And this is a snare. Irma, you had a question? Complex, yeah. Yeah. A SNAP and T-snares are usually very closely associated membrane. They have an affinity for each other. And so when this V-snare encounters a T-snare, it almost never encounters it alone. It encounters it with the SNAP usually. And certain SNAPs only associate with certain T-snares, and those SNAP T-snare complexes only associate with specific V-snares. That's why those vesicles won't go fusing someplace else. They can only fuse with that basement membrane where the synapse is, because that's where the T-snare SNAP combo is. So it's all very specific. The cell doesn't leave this to chance.
And the specific vesicles that contain specific neurotransmitters?
Yep.
Or so each neurotransmitter
Yep.
vesicle has different.
Well, to keep in mind, the neuron usually is only going to make one neurotransmitter. Well, that's actually a lie. It'll make one major. I think that's fair. One major neurotransmitter. Now, if it makes another neurotransmitter, it'll have different V-snares. Yeah, it'll make complexes that will be released under certain conditions. Hey, zoomites. Zoomites. I'm going to coin that term. If you guys can hear me, unmute yourself and say so.
Yeah, I can hear you.
Okay, cool. You can? Cool. Great.
Your slides aren't moving, but I can hear you.
Slides aren't moving? Oh, good gracious. All right, let's try this again. Okay, now are they moving?
Yep.
They are good. Okay. All right. So here's the first step in this process where the T-snare, you can see, and SNAP are all very unstructured. So the snare proteins contain a repeating heptad. Heptad, that's a 7 amino acid sequence. And that heptad will associate with heptad sequences found in the SNAP and the other snare. They will then form a tight 4-helix bundled structure. 4-helix bundled structure. That looks like this, essentially. And that zipping together forces the membrane so close together that the dynamically feasible thing for it to do is diffuse. Because with this proximity up here, you got tremendous amounts of ionic repulsion, but these snare bundles are going to continue to pull it together. It's kind of like, oh boy, that's a really dumb example. No, I'm not going to use that. That's a really stupid example. Oh, yeah. Okay. Football. Those of you who played football. Who played football here? Okay, good. So if you're on the offensive line and it's a short yardage situation and the runner hits the line, sometimes you leave your blocker, you get behind the runner and you push on the runner. And the whole pile just kind of goes kind of forward like that because you're behind pushing. And that's kind of what happens here. The runner has to go forward because all these big meat market people are pushing behind him, essentially. And I always played defense, so I never had the pleasure. I used to always just get on the ground and make everyone trip over me. That way, the play would just go like that. They hated me. Anyway, that's kind of the way this works. Because they're so close, the repulsion is so grand, but the force pulling them together is much stronger. The fusion is thermodynamically the synergy thing for them to do. So these membranes will fuse. And then you have, first, you have the miniature fusion like this, the so-called hemifusion. And then this is not feasible because you have repulsion here and here. This then fuses because this is going to stay fixed like that. And now you have the formation of a complete fusion pore. And as the pore widens, the vesicle contents then will be released.
What happens when you have the kiss and run type when you don't actually collapse? Is it the same process? What causes it to be the collapse? Just open a little bit and then back up.
If the membrane is particularly stable under certain conditions and you get to hemifusion, sometimes it may go back the other way to that. And then these will get unwound because there are proteins that unwind these. And then you're back to where you started, essentially. You have to try it. Usually the membrane has enough movement so that this situation is not at all stable. If, however, under situations the membrane is pretty stable, like cold temperatures, for example. In fact, if you work with acinar cells in culture and you want a sound, great way to do it is to pick the whole thing up and take your experiments into the cold storage room. And that stiffens the membranes, and now this process isn't nearly as efficient, and that slows secretion down a whole bunch. And that's one example where you could do that, where that would happen.
Yes, but when this process is over, then when your neurotransmitter gets what happens to the core helix complex?
Oh, we're getting to that. Okay. We're getting to that. So that 4-helix complex, oh, and additionally, I didn't mention, membrane fusion is also driven by the hydrolysis of GTP by this Rab protein. That gives it an extra oomph that essentially drives this fusion in the forward direction. Andrew? Yeah, the membrane gets bigger and then pieces of it get recycled. So there's a constant recycling of membrane.
Is it in the same place or does it recycle from a different part of the membrane?
Recycling tends to occur in a different part. It tends to be spatially separated. If you look at cultured neurons, in fact, if you label them with dye, you'll notice that the holes that appear after you get the neurotransmitters, holes tend to appear in about the same place. And recycling tends to occur in about the same place. So it tends to be spatially segregated. Okay, what then do you do with that complex? There is now a complex of proteins. There's a complex that's called a alpha SNAP NSF complex. Alpha SNAP NSF and NSF forms a hexamer, and then alpha SNAP forms a little base, and this has a hole in the bottom, and this thing goes down. The NSF is an ATPase, and it literally hydrolyzes ATP and goes like this, and it unwinds it. And then the V snare gets segregated and then packaged to another vesicle and sent back to be reloaded with neurotransmitter. The process repeats itself.
Where is the snare in that whole complex?
The V snare, and so you get the 4 helix. So you have the SNAP that's got, then you have the V snare, you have the T snare, like that, and that essentially just sits on top, and it literally it rotates and it goes on and it unwinds them.
That's
So
the NSF part.
that's the NSF, and sits on top and then goes like that. So the alpha SNAP, that's the soluble NSF attachment protein. That's why it's called alpha SNAP. A lot of this was discovered, well, elucidated by a screen that was done by Scott Emmer when he was a postdoc in Randy Sheckman's lab at UC Berkeley. And then Emmer got his own lab at Caltech. And Emmer then took that screen and just greatly increased our knowledge of the types of proteins involved with the Golgi and vesicle fusion, and all this was done in baker's yeast. But it turns out the proteins discovered in baker's yeast were orthologous to proteins in our cells. And now we're discovering that mutations in the genes that encode many of these proteins actually contribute to certain genetic diseases. And so this basic research in the lowly baker's yeast now is paying off dividends for human medicine. So President Clinton had a vice president named Al Gore. Al Gore was prone to say incredibly stupid things. Not prone, he is prone. He still says stupid things. But he used to say, we don't need any more basic research. We just need to apply the research we had. On the stupid scale, that's over 10. That's like 52. So on a 1 to 10 scale, that is a 52. So don't knock basic research, because that's where your applied research come from. That's where your drugs, your cures, your diagnostic things all come from, is basic research. So when someone says that, just smile and politely walk away, because they have nothing to teach you. Spondyloepiphyseal dysplasia. Spondyloepiphyseal dysplasia is a bone problem. You don't grow bones properly because the osteoblasts that release these vesicles filled with collagen, upon which hydroxyapatite will precipitate. That doesn't happen properly, so you get crooked bones. You have other problems too, but the big problem is the bones, and that's one of them. And there are several others we could talk about as well. So yeah. And there's the process as a whole. Very, very interesting topic. There's lots of work that's been done on this. If you're interested in this, again, I can't recommend highly enough the papers by Randy Sheckman Scott Emmer, that's EMR, Scott Emmer. And then if you're interested in a more in vitro system, biochemical system, John Rothman has worked extensively in this. Rothman tends to take a little data and make very grand statements about it, but those are the people whose papers you could read about if you're interested in that. All right, let's go about transport. So transport of entities across members is described by the laws of concentration and dilution. I say laws because they describe. Some people say these laws, they drive these phenomena. No, they describe them. It is the glory of God to hide things. It is the glory of mankind to discover them. You think you know more than God does? Do you think by your description you make them happen? Boy, gang, that's hubris. That is hubris on a gigantic scale. I hope you don't have that kind of hubris. Hope that when you approach the investigation of God's creation, you do so with humility. So diffusion refers to molecules in a high concentration moving and going to areas of lower concentration. This makes intuitive sense just based on the thermal movement of things. And we have a little, well, it's more of a cartoon sort of thing. Let me just show it to you. Okay. So here is diffusion. Let's just go next. And you can see we have a lot on one side, don't have much on the other. They'll randomly move, they'll bounce into the wall, blah, 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 blah. Here we go. Next. And then if we then have them start moving across that membrane, that is essentially this process called net diffusion. And we can see we reach equilibrium when all these molecules are essentially the same concentration in the same place. This was illustrated to me really very plainly when I was a freshman at UC Davis. Donald McQuarrie was my chemistry prof. If you've had PChem with Tom Kunzelman, he loves Donald McQuarrie's book on quantum mechanics. That was my freshman chemistry teacher at UC Davis, was Donald McQuarrie. And he took a canister of hydrogen sulfide gas, which smells like rotten eggs, and he opened it up and sat down, and then just row by row, ooh, yuck, terrible, rotten eggs, hey, who cut the cheese? So on and so forth, all the way back to the room. And that beautifully illustrated diffusion. Things going from an area of high concentration, then moving into areas of concentration. Because hydrogen sulfide is relatively pungent, you can smell it relatively easily. That's a nice illustration of that. Now, does diffusion occur in biological systems? Yes, but you have this thing called the membrane. That's going to complicate matters. Some things move through membranes via diffusion very easily, like gases. Other things, like water, will also move via diffusion rather easily. Most of the other things we'll look at will not. They have to move through membranes. That is a more complicated process called osmosis. Okay, if the temperature is raised, what happens if the temperature is raised? So I guess we're supposed to click on that. Does it go faster or slower if the temperature is raised? Faster, because diffusion depends upon thermal movement, doesn't it? You increase thermal movement, you increase diffusion. Congratulations. Oh, I'm so proud of myself. All right, let's get back to our PowerPoint. Okay, stop sharing. Good grief. Okay, so slides moving, everyone. Zoomerati, are the slides moving? Hello?
No, they're not moving.
No, they're not moving. Of
No, we're
course,
still on the page with the link.
still on the page of the link. Okay, let's try this again. Okay, try sharing that again. Okay, now they're moving.
We're on the osmosis slide. I don't know if you're switching between slides.
I am, they're not moving.
Nope, we're still on the osmosis slide.
Nope, then we'll just keep it here.
Okay.
All right, so if you have the movement of charged entities, that means you're going to develop a disparity of charge on one side of the membrane relative to the other. This will create a situation called a membrane potential. Membrane potentials, fortunately, can be measured. In fact, if you have a lot of dexterity, you can measure them relatively easily. How do you do this? You stick an electrode in cells. Now, with big cells like Xenopus oocytes, that's actually pretty easy to do. Problem is you got to make the bloody electrodes. That's hard. But taking these with a good microscope, you can just insert it in and you can measure the membrane potential. You can do this for a variety of cells. All cells have a membrane potential, all of them. Human cell? In culture, yeah. Because you have to get it under a microscope to be able to do this. And some human cells are really small, really hard to do. Some human cells are a lot bigger, a lot easier to do. Could I just walk up to you and do that on your skin cells? No, because your skin cells mostly aren't alive. But I have to wrestle you down and find a good cell to do it? Yeah, probably. I don't think you'd like that very much. You'd probably resist too, so I probably wouldn't do that, so no. Plus, we're friends, right? Yeah, okay. So I don't think I'd want to risk that, so yes, you could, but I wouldn't recommend it. Yeah, I have scientific curiosity, but I don't think I'd want to take it that far. So membrane potentials are pretty universal. They're not just something you see in human cells, you see them in plant cells, you see them in animal cells, and yes, we even see them in bacteria. They're very, very common. And they're due to the fact that you have a difference in concentration in charged molecules across the membrane. Now, this membrane potential can be used to do work. And it often is used to do work. And if you look essentially at this difference in concentration of your various charged entities, you can actually calculate what energy is available for work. Now, the movement of solutes across membranes will depend upon several factors. First of all, it's going to depend, let's see if we can fix that at all. Not really. So first of all, it's going to depend upon the concentration difference on the side of the membrane. The greater the difference, the greater the tendency you'll have to bring things across the membrane, into the cell, whatever. Secondly, what is the ratio of positively charged ions inside and outside the cell? What's the charge differential? You may have a charge differential that has little to do with your ion, but it may help you bring your positively charged ions inside the cell if you have a disparity. There's a third feature that actually is not mentioned here, and that is how permeable is the membrane to your ion. That's a factor too that you're going to need to take into consideration. So if we consider things like calcium ions, sodium ions, potassium ions, protein, because proteins are charged, chloride ions, whatever, and we sum all those together, we get this consideration that takes into account for all the concentration differences and the charge differences, and this composes a so-called electrochemical gradient, otherwise known as the electrochemical potential, because that energy is the potential to do work. And by work, we transport work, usually across the membrane. And this is the cumulative effect of these differences in concentration across the membrane. Now, we'll talk about this a little bit later, but your book goes into this equation called the Nernst equation. And the Nernst equation is the mainstay of neurophysiology, and it can be used to calculate that membrane potential. I'll talk more about that at another time, but the Nernst equation is how you go about calculating this. So when it comes to transporting things across the membrane, some things don't need transport. So membranes, biological membranes, are selectively permeable. I guess you could also say they're selectively impermeable, but we're thinking of the glass being half full rather than half empty here. So small polar molecules like water, they get diffused through the membrane. Small nonpolar molecules like benzene, steroids, naphthalene, things like that, they can diffuse through the membrane. Larger polar molecules like glucose, other carbs, no. Too big, too polar. Ions, sodium, potassium, calcium, no. Too charged. Gases, yes. Non-polar gases, very quickly. Polar gases, not quite as quickly, but quickly enough. If you want to get solutes into the cell, you usually need some sort of carrier. That carrier can be a transporter, or it can just be a pore. Pores, remember, number one, whether or not pores are open will be regulated. And pores, if they're open, they're going to respond to concentration gradients. So if a pore is open and you have a lot inside the cell, very little outside the cell, your whatever that molecule is specific for that pore will exit the cell. If it's the opposite, then it will enter the cell, and so on and so forth. Most of your solutes, this is really important, and this might be something you've heard of before. I was blessed with really big ears. Probably because I'm so darn blind. Most solutes don't exist naked in anything. In the test tube, in cells, in interstitial fluid, in blood, they're hydrated. And did Dr. K in chemistry go over Stokes radiuses? No? A Stokes radius refers to the molecular radius of a entity. The Stokes radius refers usually to the radius of it while it's hydrated. So this is, and I've tried to explain this to people online, and I usually just give it 3, 4, 5 tries and then I quit because life's too short to try to explain certain things to certain people who already made up their mind. I had somebody tell me, well, the pores in your mask are a lot bigger than a virus. Now, when we exhale, are we going to exhale naked viruses? No. We exhale, when they're big, they're called droplets. When they're smaller, they're called aerosols. Big droplets dehydrate to aerosols, dehydrate to really small aerosols. Small aerosols stay afloat for a long time, but masks block big particles that prevent them from becoming smaller particles. I tried to explain that to somebody once online. I as well have tried to explain that to this table. It did no good whatsoever. But just keep that in mind. We don't exhale naked bacteria, naked viruses. They're always hydrated. Same thing with particles. So if a particle is going to go into the cell, it has to be unhydrated. That takes energy. That's the reason why getting things through biological membranes is thermodynamically unfavored. Transports will take the dehydration energy and will drop it like a rock, so that now transport becomes much more favorable. And it does that because of the very unique fit between the transporter and the entity, and specific binding between the transporter and the entity of interest. And we'll look at a few examples of that. All right, so this is a little classification of the different transporters. So there are channels. Channels are just doors in the membrane. They're specific doors, but they're doors. They're openings in the membrane, and whether or not they're open or shut is regulated. Carriers. Carriers can be primary active transporters or secondary active transporters, or they can be non-active transporters. They can be facilitated diffusion transporters. There. So facilitated diffusion that didn't look right. These respond to concentration and concentration only. These will bring things into the cell or kick things out of the cell in a concentrated fashion. If there's a lot of it outside the cell, very little bit inside the cell, in it goes. So this works quite well for glucose. Glucose gets in the cell, gets metabolized like that. Concentration of glucose in the cell is really low. So this works really well for glucose. It'll also work pretty well for some amino acids. Your body tends to use this just for glucose. Other organisms use it for other things. So this does not require the expenditure of energy because the energy is already present in the concentration gradient. So here is the facilitated transporter from red blood cells, from erythrocytes. You could see this has 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. 12 different hydrophobic alpha helices. Those alpha helices sit in the membrane and it forms a pore just like this. And that pore essentially will accommodate the glucose quite well. Yeah.
So do pores only allow specific things for it?
Yep. Facilitated diffusion transporters are specific, very specific. The Hexose facilitated diffusion transporter in yeast will transport glucose or mannose or galactose. Yeah. There's another transporter for sucrose. And this is a model for this particular transporter. And you have the binding of glucose. You have a conformational change. That conformational change then will convert the molecule into something into which does not have a high affinity for glucose, but it will release it, but it releases it to the other side of the membrane. Once it's not bound to glucose, it will snap back instantly to this, and this cycle repeats itself. Kinetically, you can trace facilitated diffusion because it has a set of reactions that look alike, and a lot like an enzyme catalyzed series of reactions. So this will follow Michaelis-Menten kinetics. And it'll follow Michaelis-Menten kinetics quite nicely. And this essentially is your equation. You could use this essentially to determine if your entity is taken up by facilitated diffusion. Because if you map this via this equation, you should get a straight line. If you get a straight line, it follows Michaelis-Menten kinetics. You'll get a KT. That KT is very similar to the Michaelis constant. It equals that concentration at which the rate at which you take it up is equal to 1/2 the V max. And you get a V max as well. There's a point at which increase in the concentration won't increase the transport. Here is an example of the glucose facilitated diffusion transporter from erythrocytes. You could see you get a very nice hyperbolic curve and then a nice linear curve using a Line-Weaver-Burk plot. What about those transporters that utilize energy, but they don't utilize energy in the form of ATP? Instead, they utilize energy found in those concentration disparities. These are called secondary active transporters. Secondary active transporters. These will tend to transport two molecules together, or they'll transport molecules in opposite directions. And so these are antiporters or exchangers. Yes?
TeVs.
because this is utilizing energy, but it's the energy stored in concentrated radiance, and it's using it to bring something else into the cell or to kick something else out of the cell. The cell-to-tape diffusion just brings something in with a concentration gradient. So it's just bringing one thing in. This uses the energy in those gradients to bring something in or take something out. So this is the chloride bicarbonate antiporter, or chloride bicarbonate exchanger. And it will go one of two directions, depending upon if it's in the lungs or if it's in the tissues. So carbon dioxide is used by the enzyme carbonic anhydrase to make carbonic acid. Carbonic acid just joined to hydrogen ion plus bicarbonate. In the lungs, because you're giving CO2, you need essentially, you're giving up CO2 and you need to be able to disjoin that CO2 is stored in the form of carbonic acid. So you're bringing bicarbonate in to disjoin it so that you can give up that CO2. So your bicarbonate is high outside the cell, and you want to get rid of chloride because you've loaded up with chloride in the tubes. So you'll pump it this way, essentially. Bicarbonate in, chloride out. Then when you get to the tissues, it's the opposite. Now you essentially want to kick bicarbonate out because you're taking up bicarbonate as a metabolic waste. And so you're going to store that in carbonic acid, but you make bicarbonate that will help buffer the acid you make via metabolism. And now you bring chloride in. And this is called the chloride shift. Chloride is brought in the tissues, and it is removed in the lungs. So this one will switch because it depends on where the energy is. Is it on the inside? Is it on the outside? Depends where you are.
Am I correct to say secondary active transporters always transport more than one thing?
Usually, they're usually co-transporters.
You said that they're usually importers though, but you also said that they transport.
They can be symporters as well. They can bring things in together. So here are your co-transporters, your secondary active transporters, antiporters, just like the bicarbonate chloride antiporter. And supporters, for example, in the kidney, or also in the GI tract, you have the glucose-sodium symporter. And the glucose-sodium porter brings sodium and glucose into the cell. And that is actually a target for a new group of anti-diabetes medicines. They inhibit the SGLT1 and 2 in the kidney. Therefore, they cause you to lose more glucose in the urine. They drop blood glucose. They also drop blood volume, so they drop blood pressure as well. And these drugs have been shown, the so-called flozins, empagliflozin, canagliflozin, your mother-in-law flozin, so on and so forth. These drugs have been shown to decrease heart attacks in people with diabetes and heart conditions. What's a trade name you might have heard of? Invokana, Jardiance, those drugs. You don't know what the first thing I'm talking about, do you? Okay, well, anyway. So, active transporters require the input of energy. That can be in the form of ATP or the energy stored in gradients. Your primary active transporters use energy in the form of ATP. So direct hydrolysis of ATP brings things into the cell. And we can calculate using this equation. We'll do this on Friday. So your quiz for Friday. Yeah, I'll be over chapter 11. Chapter 11. And if someone is missing a tooth, gotcha, gave me this as a present. So if it's your tooth, do please let me know. It's part of the tooth, and it looks to me like it's a molar.